I want you to imagine a microscopic war happening inside your cells right at this very second.
Oh, it was a brutal war, yeah.
Right. It's this intense battle of infiltration, deception, and survival. And the enemies in this scenario are orthoflaviviruses. We're talking about pathogens like Zika, Dengue, and West Nile virus.
Yeah, these are viruses that cause devastating severe diseases globally.
And when they attack, our medical arsenal is basically empty, right? Like, we currently have absolutely zero approved antiviral treatments for them.
It's a sobering reality. For decades now, we've just been entirely reliant on our immune system to fend off these highly evolved invaders.
Which is terrifying.
Right, because the virus mutates, our immune system adapts, and the cycle just continues. But the thing is, our cells aren't just passive battlegrounds waiting for the immune system to finally show up. They actually have these hidden, built-in countermeasures that we are, frankly, only just beginning to understand.
Okay, let's unpack this, because that hidden countermeasure is the entire mission of our deep dive today. We are looking at a brilliant piece of newly published research that uncovers a completely unexpected cellular weapon, a host protein called SPART.
It's an incredible discovery.
It really is, and we've got a massive stack of sources to go through today. We are talking about intricate, genome-wide CRISPR activation screens, complex molecular biology assays tracking glowing viral RNA, and even live mouse models. Our goal is to figure out exactly how the SPART protein manages to trap these viruses and, what this discovery means for the future of medicine. But to understand the defense, we kind of need to understand the break-in first.
Yeah, let's trace that offensive playbook.
Yep.
So, an orthoflavivirus, like Zika, first attaches to the outer surface of a human cell.
Okay.
The cell then basically swallows the virus up in this process called clathrin-mediated endocytosis.
Clathrin-mediated.
Right, so think of clathrin as a sort of molecular scaffolding. It builds up on the inside of the cell membrane, pulling it inward until it pinches off a piece of the membrane to form a tiny internal bubble.
Oh, wow.
Yeah, and this newly formed bubble carrying the virus is called an endosome.
So the virus is inside the cell now, but it's essentially trapped in a quarantine room. Like, it's a Trojan horse, but the horse hasn't actually been allowed into the city yet.
That's a great way to look at it. So as this early endosome matures into what we call a late endosome, the environment inside that bubble becomes increasingly acidic.
Acidic, why?
The cellular machinery physically pumps hydrogen ions into the bubble, causing the pH to drop drastically. And this acid is the exact trigger the virus has been waiting for.
So the quarantine room turns into a trap, but the virus actually wants that.
Exactly. The low pH causes a massive shapeshift in the viral envelope proteins, forcing the viral membrane to fuse directly with the endosomal membrane. But there is one crucial final step before the virus can actually take over the cell and start copying itself. It's a step called uncoating.
Uncoating. Because the virus's genetic material, its RNA is locked inside a really tough inner shell called the capsid, or C protein, right?
The capsid.
It reminds me of, like, a coat check at a highly exclusive club. The virus has managed to sneak into the secure lobby that's the endosome, but it is wearing this heavy, super conspicuous coat, the capsid.
I love that analogy.
So to get past the inner doors and enter the main party, the cell's cytoplasm where it can start multiplying it, absolutely must take that heavy coat off.
That visual captures the mechanical problem perfectly. The genomic RNA is effectively inert while it's encased in that capsid protein. It cannot interact with the cell's ribosomes to make new viral proteins. It has to be released.
But wait, if the RNA is completely inert inside that shell, how exactly does the virus know how to take its coat off? I mean, a virus is just a tiny packet of genetic instructions. It doesn't have hands.
Right.
Does it rip the coat off all by itself, or is our own cell somehow helping it disrobe?
That was the exact mystery researchers needed to solve. And to find out, they didn't focus their microscopes on the virus itself. They looked at the host cell. They set up something called a genome-wide CRISPR-Cas9 activation screen.
Let me stop you there, because usually when we hear about CRISPR, we think of molecular scissors, right? We think of scientists cutting defective genes out of a DNA strand. But this is an activation screen.
Yeah, the mechanism here is beautifully modified. They used a system called dCas9 VP64.
Okay, dCas9.
The D stands for dead. They essentially mutated the Cas9 protein so that its molecular scissors are completely broken. It can't cut DNA anymore.
Oh, interesting.
Instead, it just acts as a highly precise GPS delivery truck. It finds a specific gene, parks there, and delivers a payload called VP64.
And what does VP64 do?
It's essentially a molecular volume knob. When it arrives at a gene, it cranks up the transcription machinery, forcing the cell to produce massive amounts of whatever protein that gene codes for.
So instead of deleting genes to see what breaks, they are turning up the volume on all 20,000 plus genes in the human genome, one by one, to see what gives the cell a superpower.
Exactly. They used human placenta cells, specifically a line called JG3, which are highly vulnerable to Zika. They turned up the genes, infected millions of these cells with a massive, lethal dose of Zika virus, and then just waited.
Sounds brutal.
It is a brutal selection process. The viruses wipe out almost everything in the dish. But then they extracted the DNA from the few surviving cells to see which gene's volume had been turned up.
So what did the survivors have in common?
Well, several genes were overexpressed. Some were known antiviral defenders, like genes called IFV6 and AHSP. But one hit really stood out to the research team, a gene called SPART, which produces a protein called spartan, or SPG20.
Sartan, okay.
What made SPART so remarkable was its potency. Its expression only needed to increase by a mere 1.64-fold to severely crash the virus's ability to replicate.
Wait, really? Just 1.64?
Yeah. The cell didn't need to be blasted with 1,000,000 times the normal level of this protein. Just a tiny bump in SPART was deadly to the Zika infection.
Wow. Okay, so they found this incredible shield. But how did they figure out where SPART is fighting the virus? Because cells are massive, complex environments.
They used a brilliant process of elimination, utilizing specific chemical inhibitors to freeze different parts of the viral life cycle.
How does that work?
First, they introduced a chemical called cyclohexamide, or CHX. This chemical binds directly to the cell's ribosomes, completely jamming the gears of the protein factory. It stops the translation of RNA into proteins dead in its tracks.
Okay.
When they treated the cells with this chemical, they found that the SPART protein still successfully lowered the levels of viral RNA.
Oh, I see. If you completely freeze the virus's ability to manufacture new proteins, and SPART is still destroying the virus, that proves SPART's attack has to happen very early on. Like, it's hitting the virus before translation even starts.
Exactly. Next, they deployed a different chemical called borfamycin A1, or BAFF-A1. This chemical is a proton pump inhibitor. Remember how the endosome bubble needs to become acidic to trigger the virus?
Right, the trap we talked about earlier.
Well, this chemical stops the cellular pumps from pushing acid into the bubble, which completely paralyzes the virus. It can't fuse, and it can't uncoat. When researchers compared these chemically paralyzed cells to cells that simply had extra SPART protein, the results were biologically identical.
So SPART is acting like that chemical. It's trapping the virus in the bubble.
Yeah, and to confirm this beyond a shadow of a doubt, they utilized an imaging technique called a pi-Fisher assay fluorescence in situ hybridization.
Sounds complicated.
It sounds complex, but essentially they engineered glowing molecular probes that bind exclusively to Zika's RNA. Under a high-powered microscope, the viral genetic material literally lights up.
Yo, that's cool.
In normal cells, you see this glowing viral RNA spread diffusely all throughout the cytoplasm. The virus successfully took off its coat and infiltrated the party.
And in the cells with the boosted SPART.
The glowing viral RNA was tightly clustered. It was entirely trapped inside specific bubbles marked by a cellular protein called Rab7.
Rab7?
Rab7 is the biological street sign for late endosomes. So the imaging proved definitively that SPART traps the virus inside the endosome. The virus couldn't take off its heavy coat.
Okay, let me make sure I'm wrapping my head around this. Let's do a quick recap for you, the listener, since we are about 5 minutes in.
Good idea.
Bullet 1, viruses like Zika sneak into cells via these bubbles called endosomes. But to survive, they absolutely must shed their heavy capsid coat.
Right.
Bullet 2, a massive CRISPR volume knob experiment revealed that slightly boosting the SPART gene traps the virus specifically inside the Rab7 coat check room.
Exactly.
But this raises a huge question for you to think about. If SPART is trapping the virus, is it attacking the virus directly like a targeted missile, or is it doing something far more deceptive?
What's fascinating here is that SPART does not touch the virus at all.
Wait, really?
Not at all. When researchers tried to force the SPART protein and the viral capsid protein to interact in a controlled lab assay, there was zero biochemical connection. They actually repel each other.
So how does it stop it?
SPART is actually targeting an unwitting accomplice inside our own cells, a traitor.
Who is the traitor helping the virus?
It's a host protein called ITCH. ITCH is an E3 ubiquitin ligase.
Ubiquitin ligase. What does that mean for the cell?
In normal, healthy cellular life, ITCH is essentially a sanitation worker. Its job is to roam around finding cellular garbage misfolded or old proteins we don't need anymore and tag them with a tiny molecule called ubiquitin.
Okay.
Ubiquitin is basically the biological kiss of death. Once a protein is tagged with a chain of ubiquitin molecules, it is sent to the proteasome, which operates like a microscopic wood chipper.
A wood chipper.
Yeah, the tagged protein gets shredded into basic amino acids to be recycled.
So how does Zika exploit a cellular sanitation worker?
Well, the researchers discovered that the Zika capsid protein has evolved to intentionally interact with ITCH. Specifically, a single amino acid on the Zika capsid, a lysine at position 5, or K5, presents itself directly to ITCH.
Wait, so the virus is asking to be tagged?
Yes. ITCH just does what it's programmed to do. It sees the K5 residue, tags it with a ubiquitin chain, and the cell's proteasome dutifully shreds the Zika capsid. For dengue, by the way, it's a neighboring residue called K6, but the trick is exactly the same.
Here's where it gets really interesting. Going back to our club analogy, ITCH is the coat check attendant.
Yes.
The virus is wearing this heavy capsid coat that it cannot take off by itself. So it deliberately tricks our own cellular sanitation worker into ripping the coat off and throwing it in the shredder. By destroying the capsid, ITCH is unwittingly helping the virus release its naked RNA into the cytoplasm.
It is a brilliantly devious viral strategy. The pathogen relies entirely on our own cellular machinery to complete its break-in. But this is exactly where SPART executes its masterstroke.
Okay, tell me about SPART's move.
If you look at SPART's molecular structure, it features a very specific shape called a PPA-Y motif that's a sequence of 4 amino acids: proline, proline, alanine, and tyrosine.
PPA-Y.
Right. SPART uses this PPA motif like a grappling hook. It binds incredibly tightly to ITCH and physically drags the sanitation worker away from the Rab7 late endosomes, scattering it diffusely all throughout the cytoplasm.
Oh my gosh, SPART kidnaps the coat check attendant. It pulls ITCH away from the endosome lobby, so ITCH just isn't there when the virus arrives.
Exactly. Without ITCH present at the late endosome, the viral capsid never gets tagged with ubiquitin. The proteasome wood chipper just ignores it.
So the RNA is stuck.
Right. The viral RNA remains permanently locked inside its capsid shell, trapped inside the acidic endosome. And eventually, the cell realizes this bubble contains useless, degraded material and safely digests the entire package.
It's literally a microscopic chess match. But how did they prove that dragging ITCH away is the specific move that wins the game?
They engineered a mutated version of SPART, which they called SPART-M3. This mutant was identical to the normal protein in every single way, except they deleted that specific 4 amino acid PPA-Y grappling hook.
Oh, so it couldn't grab ITCH anymore.
Exactly. Without the PPA motif, the mutant SPART couldn't grab ITCH. And without grabbing ITCH, the mutant SPART completely failed to stop the Zika virus. The virus uncoats and replicates just fine. It proved that kidnapping ITCH is the exact necessary mechanism of the defense.
That is wild, but we have this pristine molecular mechanism working beautifully in human placenta cells in a petri dish. Biological systems in a dish don't always behave like a living, breathing organism with a complex immune system, right?
Yeah.
Does this coat check kidnapping actually protect a living creature?
To answer that, the research team had to move to in vivo studies using live animal models. They utilized immunocompetent pregnant mice.
Why pregnant mice specifically?
Because one of the most devastating impacts of Zika is congenital Zika syndrome. During vertical transmission, when the virus passes from the mother through the placenta to the fetus, it leads to spontaneous abortions, microcephaly, and severe neurological abnormalities. They really wanted to see if this mechanism protects the next generation.
How did they set the experiment up?
They bred 3 distinct groups of mice. The first group was genetically normal, wild-type mice, or WT. The second group was genetically engineered to completely lack the SPART gene, meaning they had no SPART protein at all. They call those SPG20 knockout mice.
Okay, and the third group?
The third group was engineered to lack the ITCH gene. Then they injected all the pregnant mothers with a massive uniform dose of Zika virus.
Let's start with the baseline. What happened to the normal mice?
In the normal wild-type mice, the infection was severe. About 40% of the fetuses were affected by congenital Zika syndrome, showing extreme growth restriction or fetal resorption.
Fetal resorption.
Yeah, which is when the embryo is essentially lost, leaving only remnants of the placenta. And when they measured the viral RNA copies, they found high viral loads in both maternal spleens and the placentas.
40% affected is devastating. So what happened to the mice engineered without the SPART gene, the ones missing their cellular shield entirely?
It was catastrophic. Lacking SPART made the infection aggressively worse. The researchers found significantly higher viral loads in the maternal brains and spleens, as well as an absolute explosion of viral replication in the fetal brains compared to the normal mice.
Because they had no defense.
Right. Without SPART to kidnap ITCH, the virus had completely free rein to use the coat check attendant everywhere in the body, uncoating and multiplying without restriction.
Which brings us to the real test. What happens when you remove the traitor? How did the mice lacking the ITCH protein fare?
The results were striking. When the unwitting accomplice, ITCH, was completely removed from the equation, the mice were highly protected. Only 21% of the fetuses were affected, nearly cutting the casualty rate in half compared to the normal mice.
Wow, that's a massive drop.
Yeah, the viral loads in the spleen, placenta, and fetal brains completely plummeted.
Yeah.
Because ITCH simply wasn't there to uncoat the virus, the pathogen couldn't establish a strong infection, which ultimately saved the majority of the pregnancies.
Okay, let's do another quick recap for the listener since we're about 10 minutes into the deep dive now.
Let's do it.
Bullet 1, the virus tricks our own protein, ITCH, into destroying its capsid coat so it can infect the cell. Bullet 2, SPART, saves the day by physically dragging ITCH away from the virus using a PPA motif grappling hook. And bullet 3, live mouse models prove that deleting ITCH actually protects against the virus, while deleting SPART makes the infection devastating.
Spot on.
Which brings me to a big question for you listening at home. This dynamic works beautifully for Zika, but there are thousands of different viral families out there causing pandemics. Does SPART work on all of them, or is this a highly specific weapon? Because I have to push back on this a little bit with you, our expert.
You go for it.
If SPART is this incredibly effective natural shield that saves fetuses from a devastating virus, why isn't our body just cranking up SPART production all the time? Is there a downside to just flooding our system with this protein?
That is a crucial biological point. You really have to remember ITCH's day job. It is a vital sanitation worker. Our cells constantly produce misfolded proteins and metabolic waste that need to be cleared out by the proteasome.
Right.
If our bodies permanently overexpress SPART, it would constantly kidnap ITCH, sequestering it away from its normal everyday duties. Cellular trash would build up rapidly, leading to toxicity and cell death. The system relies on a delicate, temporary balance. SPART is a targeted weapon, not a permanent state of being.
That makes total sense. You can't permanently lock up your sanitation workers. Which leads me to wonder about the scope of this weapon. Like we just asked the listener, does SPART work against other viruses? Could we use this exact mechanism to fight something like COVID-19 or herpes?
If we connect this to the bigger picture, the short answer is no. When they tested SPART against SARS-CoV-2, herpes simplex virus type 1, and an alpha virus called Synbis, it did absolutely nothing. The viral replication for those pathogens was completely unaffected whether SPART was present or absent.
Why does it only work on Zika? What makes Zika so special compared to a respiratory virus like COVID?
Well, it's not just Zika. SPART strictly restricts an entire family of pathogens called the orthoflaviviruses. This includes Zika, Dengue, West Nile virus, and Japanese encephalitis virus, or JEV.
Okay, so it works on all of those.
Yeah, when they infected mice with Japanese encephalitis, for example, the mice lacking SPART suffered rapid weight loss and high mortality, while the mice lacking ITCH survived. So SPART acts as a pan-orthoflavivirus shield. And the reason it only works on this specific family comes down to cellular geography.
Geography inside the cell.
Yes. The researchers used advanced 3D confocal microscopy to map out exactly where these different viruses go once they get inside.
And what did they see?
It turns out that only the capsid proteins of orthoflaviviruses naturally localize to the Rab7-positive late endosomes. That specific Rab7 endosome is the doorway they use, and that doorway is exactly where ITCH operates to uncoat them.
Oh, I see. It's all about location, location, location. If a virus doesn't use that specific coat check room, it doesn't matter if SPART kidnaps the attendant there.
Exactly. Other viruses, like the Synbis alpha virus or SARS-CoV-2, use completely different entry pathways. They fuse at the cell surface or use different types of internal bubbles. They don't need ITCH, and therefore SPART's grappling hook tactic is utterly useless against them.
That is fascinating.
Interestingly, the researchers also mapped the specific ubiquitination sites for other orthoflaviviruses. Like I mentioned earlier, while Zika uses the K5 residue on its capsid to trick ITCH, Dengue uses a neighboring residue called K6. But the mechanism tricking the sanitation worker to shed the coat remains identical across the whole family.
So what does this all mean? Even though it's highly specific to orthoflaviviruses, finding a single cellular choke point for dengue, Zika, and West Nile is a massive leap for drug design. We started this deep dive talking about how our medical arsenal is empty, zero-approved antiviral treatments for these diseases. Now we have a biological blueprint showing us exactly how to disrupt their life cycle.
We do. We have uncovered a deeply elegant mechanism. The virus relies on the Trojan endosome. It orchestrates an unwitting betrayal by ITCH to remove its capsid. And in response, the host cell utilizes SPART's highly targeted intervention to kidnap ITCH, permanently trapping the virus inside its own entry bubble.
It radically changes how you look at a viral infection. It's not just a virus kicking the door down and overwhelming a passive cell. It's an active, dynamic struggle, utilizing our own proteins against us, while our cells fight back with incredible precision.
This raises an important question, though, and perhaps a slightly ominous one for the future of drug design.
What's that?
If we take this blueprint and successfully develop a therapeutic, say, a drug that safely mimics SPART to temporarily lock up ITCH during a severe dengue or Zika infection, how might these orthoflaviviruses evolve to survive? If we permanently block their access to ITCH, could they simply mutate their K5 or K6 residues to trick a completely different sanitation worker somewhere else in our cells?
Wow. That is a profound thought to end on for you listening right now. If we block one door, evolutionary pressure might just teach the virus how to pick the lock on another. It really highlights the endless evolutionary arms race happening between our biology and the viral world. Thank you for joining us on this deep dive. Keep asking questions, and the next time you hear about a new viral breakthrough, take a second to marvel at the hidden, microscopic battles fighting to keep you safe inside your own cells.